Okay, continuing on. So we're talking about perspiration monitors over here on the bottom right side. So you have a sweat pores adhesive, then you have an electrochemical sensor chip. So this is the electronics. It's possible that this portion of the device could be reused. There's a sensor here, the electronics here, and this whole thing around the outside is an antenna. It's an inductive antenna, so the information can be read out wirelessly. Then you have a paper microfluidic device, which picks up the sweat in your skin and it brings it over to this node, these different nodes through capillary action, where the electrochemical reaction could then occur. This shows a little bit of the physiology of the body and how sweat actually is a sampling of the interstitial fluid within your body. So it's a good measure of what's going on inside. It's another example here that shows, this is an article, an IEEE article that showed a perspiration monitor where they had two electrodes like this, and this was on a sweatband. Yet another area. Again, we're just giving a very broad overview here. This one is titled Portable Imaging Technologies for Global Health. So in addition to the microfluidic aspect, the disposable part which handles the samples, which comes in direct contact with the samples, we also have to develop imaging technologies as well. So this is what would happen on the cell phone or the reader portion of it. How can we do these? How can we use cell phones and other inexpensive electronics for doing biomedical testing? Just imaging, for example. Let's see here. Some examples here. On the left here, slide A here, that's a tomographic device that could be used to image different areas of your ear and your retina. So doctors sometimes use these types of devices when they're inspecting your eyeballs or your ears in the doctor's office. They're looking at making these devices small and portable. Over here in the middle here, part C, that's an example of a device which can image inside your stomach. So there would be cameras either embedded in that capsule. You can see this capsule in the middle here right next to that penny. You can embed cameras in there, or you could have a fiber optic cable which took the signal to something on the outside, making this portion of it very much less expensive. So by inserting this device into your stomach, into your esophagus, you could image different parts of it at a reasonable cost. Then over on the right side here, we have examples of where a smartphone can be used as a microscope. A lot of types of diagnostics that would be done in a biological laboratory involves just taking cells from your body, putting them on a dish, and just looking at them. This is called morphological analysis. Okay. There's other simple experiments you can do. You can stain the cells with a certain dye, and that will cause certain areas to stain different colors. Based on that, you can make certain analyses of cells. You can do fluorescent imaging. Okay. The idea behind using a cell phone here, obviously, is that you could make the diagnostic. Maybe you could have kits that you could send to people in their homes and have their cell phones actually do the readout. Okay, so the expensive portion of it, the electronics and the reading and the transmission of the data, that could all be embedded into your cell phone. So, for example, your cell phone has a camera. Okay, the camera on the cell phones actually now, the quality of those cameras is actually much better than they used to be, right? You have a high megapixel resolution. Not just that, but the optical. I'm sorry. Stabilization. The optical stabilization is one aspect if your hand is moving, but you could also keep the cell phones situated, like you could keep it fixed. but the noise on the sensor, the signal-to-noise ratio of the sensors also has gotten a lot better. You can see that a 2 megapixel image in today's cameras, today's DSLRs, and a 2 megapixel image in like 10 years ago, it's still 2 megapixels, but the quality of the image now is much better. This is because the sensors have a much better signal-to-noise ratio than they used to. Their light capturing efficiency is better. Okay? So as a result, you can actually use your cell phone cameras today to actually do like quantitative diagnostics. Okay? It's getting to that point now. So over here on the right, you can see that there's some examples of things with an iPhone, an HTC camera, and so on, that you are actually taking images of cells. So your cell phone on its own may not be able to take pictures of cells that are, 10 micrometers in size. 10 micrometers is simply too small. But with low-cost, portable, like lenses, you could potentially use, you could attach these types of small lenses on the top of your cell phone camera, and then those lenses would serve as a microscope, allowing you to see these cells at a decent resolution. Now, I'm not going into all the details of it, but there's a lot of like tricks and techniques that you can do in order to improve the resolution of this type of imaging. For example, there's something called super resolution microscopy, where you can take pictures by using signal processing techniques. You could get pictures of cells at a much better resolution than you could as with the native resolution of the camera. Okay, this is something that I don't know too much about. I attended a talk that was on Oskin's group. He's from UCLA, and he gave a talk on some super resolution microscopy. They were using traditional CMOS sensors, inexpensive CMOS sensors, but using this technique, they could get very high resolution images. And what they were using it for was counting cells. You had a bunch of cells plated out on a chip. They could count the cells, and they could kind of get an idea of how big the cells were, so you could sort of count the different numbers of cell types just with a camera phone. Imagine that. So the example here, they had a cell phone lens, a CMOS sensor, a color filter, and so on. These are all components that are cheap now. These types of things, to make a setup like this before, would cost a lot of money. But you think about it now, you can buy the camera on your cell phone, you could buy it for maybe less than $10. That's how much the cameras cost right now. Your cell phones cost a lot more money than that. Your cell phones cost several hundred, but you figure Apple's making a lot of profit on it. The actual components that go into a cell phone are actually pretty cheap, pretty inexpensive, just because we've been making them for a while. Manufacturing economies of scale have gotten a lot better. So the CMOS sensor, the camera chip, is actually not that expensive. These types of color filters and lenses, they're made from plastics. You can make those in a relatively inexpensive manner. LEDs, the light source, also very expensive. They cost pennies nowadays. So all these electronic components that used to be expensive, which caused these diagnostic devices to all be in the hospital, now they become cheap enough where they can be distributed. Okay. So when you're thinking about your projects for this class, like if you're interested in global health, you might think of a new type of portable diagnostic. Maybe you're interested in organs on a chip. You think of like in a type of in vivo model that you'd like to explore. If you're interested in cell separation, you can think about using, making new types of cell separation devices. You see, there's a lot to explore in this field, which is why in your final projects I want to give you all the flexibility to explore whichever area that's of interest to you. Okay, and I think the rest of the class will benefit from learning about that. This particular slide, you can see, for example, it has a lot to do with electronics. It has a lot to do with imaging and optics and signal processing. And some of the previous slides had a lot to do with microfluidics, just fluid phenomena. Okay, so summing up here, BioMEMS provides several benefits, so we hopefully have hammered that into you here. Different types of small-scale benefits, microscale physics. We can exploit how the physics at the microscale. That's what the next module is going to be about. You can get higher throughput. You can decrease the cost of your device by using less reagents. You can improve quantitation by having multiple steps integrated and by taking advantage of the fact that flows at microfluidic devices are very predictable. Your devices become more portable. This next point here, BioMEMS are driving many of the emerging innovations in the biotech industry. This is a true statement. There are examples, if you look at the history of science and technology, there are examples where science ultimately led to a technology. For example, well, rather awful example is the atomic bomb, right? The development of the science of atomic scale physics ultimately led to this device, the atomic bomb. In other cases, the technology drives science. For example, in the same example, the technologies used to understand particle physics and things like that ultimately led to a better understanding. So science and technology go hand in hand. What's happening right now in the BioMEMS industry especially, these microfluidic devices, the technology is actually helping people in the life sciences do better science. One of the examples we gave was single-cell screening. Before, we didn't have the ability to do that. Now we have devices that can do single-cell, what's called patch clamping, where you can measure the membrane potentials of single cells. It's yielding a lot of new scientific insights, which ultimately will lead to better therapeutics. Okay, so this is why BioMEMS is a really, really interesting area right now to be in, both from the technologist standpoint and from a scientist's standpoint. The scientists are the ones who are using these new devices for doing new types of science. This third point here, equivalent circuit models can be used to analyze microsystems in multiple domains. This is actually part of the next module. Previous year, I actually had these two compacted into the same module. So just ignore these last 2 points right here. Scaling is an important benefit of microscale systems. We're going to talk about this in the next module, analyzing the governing equations, observing how they scale with length, and so on. So just to end here, are there any questions about this module? About just the different areas within BioMEMS, or just any questions you might have? Nothing specific. Okay. Okay. No. Okay. Let's end here then, and let's take a 5-minute break, 7-minute break, and then we'll continue with miniaturization and scaling laws in the next module.